Okay, I think I see we got our two speakers here, so we'll go ahead and get started on time today. Thanks everyone for being here. It's our April lab advisory meeting. We'll just go ahead and get started. Jeff, I think I saw you here.
Yep, I'm here.
Do you have slides to share?
I do have some slides to share.
Okay, you should be able to do that now. Or let me stop.
Okay.
Yeah, there it goes.
Can you
There it
see
goes. Yep,
it? All right, perfect. Let me just let's display the settings here. There. That look good?
it does.
All right, perfect. Well, I guess we'll go ahead and get started then. So, yes, hello everybody. You know, with spring here, that also means ticks are here as well. I don't know if anyone's had a chance to go outside and go out on any nature walks or hikes or anything like that, but the ticks are certainly out. We just did our first tick survey last Friday, and we collected a good number, over 100 ticks. So they're definitely out. They're definitely active. And so, I guess in lieu of celebrating, the start of tick season, I just want to talk a little bit about tick surveillance that we've done with our local health department partners as well as NPHL in terms of testing our ticks and that's over the last several years. So, um, before I really get started in the actual tick surveillance stuff and kind of going over what we found, just a brief kind of overview of ticks themselves. So ticks are not insects; they are actually closer related to spiders and scorpions. Adults and the nymphal stages have 8 legs, while the larval forms have 6. And ticks are what are known as obligate hematophages, so they need blood to survive and move on to the next life stage. And there's actually two main groups of ticks that are found worldwide. There's the hard ticks, which are responsible for most of the diseases that we know of, in the US as well as worldwide. And then there's the soft ticks, which can transmit certain diseases, but these diseases are typically much more lower incidence compared to the diseases that are transmitted by hard ticks. And so it's really going to be the hard ticks that we're concentrating on with this presentation. So a little bit about the tick life cycle. So ticks will emerge from eggs, so they hatch out into these six-legged larvae. These six-legged larvae will then attach and feed on their first host. They fall off, they find a place to molt, and then they emerge into these eight-legged nymphs. The eight-legged nymphs then attach and feeds on the second host. From there, they will again drop off, molt, and then emerge as adult males or females. And then from here, the males and females will attach to the third host for feeding and mating. After that, the adults will fall off. Males will just basically die. The females will go lay their eggs and then they will die. And then cycle starts over. And typically it takes about 2 to 3 years for this life cycle to be completed, dependent upon temperatures as well as the availability of hosts. And it's at these three different areas. So if you saw the green bacteria here pop up, it's these three different stages where ticks can pick up a pathogen and then from there can possibly transmit that on to humans. Typically, though, the highest risk life stages are going to be the nymphal adults or nymphal stage and then the adult tick life stage. Uh, Depending on which pathogen it is, will really, that will influence what is the riskiest life stage. So, for example, Lyme disease, it's going to be the nymphal life stage that is the highest risk for disease transmission to people. Whereas if it's something like Rocky Mountain spotted fever, it's going to be an adult life stage tick that is more likely to transmit the disease to a person. So how do ticks find a host? Well, they do not fall out of trees. I know that is a kind of a common notion that ticks will fall out of trees and get onto people. Likely this is because people will often find ticks up in their hairline, behind ears, etc. But really what ticks do is they do a process called questing. So they will hold out their front legs. They have some sensory organs that are in those front legs that they can sense different chemicals such as CO2. They can also sense vibrations coming through the ground. And so when they sense the CO2, they sense those vibrations, they'll basically crawl up onto higher onto a piece of vegetation, hold out those front legs, just waiting for something to walk by. And as that host, be it an animal or a person, walks by, they basically grab on. They got these little hooks at the ends of those front legs that allows them to get onto their host. And then from there, they will crawl onto their host and find a good spot to try to feed. Now, where are the ticks located at? Well, basically anywhere there's vegetation. Typically where we find our highest density of ticks is kind of in that vegetation that's anywhere from ankle to knee high. But it's going to be anywhere, in places such as grassland areas, forested areas, grassy areas next to kind of forested areas, or even just in the leaf litter itself if it's kind of the fall time. So, anywhere there's kind of some vegetation for ticks. I know we've even seen ticks here in Lincoln on the street corners at some of the little, just kind of little plantings of flowers and stuff that they have. There's certainly urban animals such as raccoons and foxes that run through, and certainly the ticks will fall off these animals and can actually get onto these little side areas where little gardens and stuff just downtown are actually located too. So looking at the medically important ticks in Nebraska, so there's four primary ones. When I think of important ticks in Nebraska, there's the Lone Star tick, there's the American dog tick, there's also the black-legged or deer tick, and then there's the Gulf Coast tick. Really, the three first ones there are probably our three most important ones. We're just starting to really see detections of the Gulf Coast tick here in the state, but these other three are the ones that are responsible for most of the diseases that we see transmitted in Nebraska. So we'll pretty much concentrate on those going forward. And so they're responsible for a number of different disease conditions that they can transmit, as well as some conditions that are not a pathogen. So, for example, alpha-gal and tick paralysis. These are more due to chemicals and other compounds that are in the tick's saliva that are the cause of these two conditions. So why do we even do tick surveillance? It helps us provide actionable evidence-based information to clinicians, the public, and even policymakers. It really allows us to kind of track that distribution and abundance, helps us identify pathogens that are present in the ticks, as well as quantify that prevalence. It also helps us define when ticks are active. Additionally, it can help us explain epidemiological trends, such as expanding ranges or incidence of tick-borne disease cases. It can even help us, determine the risk of exposure to tick-borne diseases that are not notifiable. So I give an example here of Borrelia miyamotoi, and in an upcoming slide, you'll kind of see why I give that as the example. And it also helps us predict the future expansion of ticks and tick-borne disease cases in the state or, in the U.S. as well. So here, the next few slides here, just going to go over the results of our tick surveillance that we've done over the last several years with our partners. And upcoming on the slides, there'll be tables that kind of show these results. And we've broken those tables down geographically based on the NOAA climate divisions. And so just for reference, here's this map to kind of show you what those climate divisions are in the state. So first, we'll just look at the tick densities here. So the first column here, we'll be looking at Amblyomma americanum. That is the Lone Star tick. And what we can see is that we have detected that tick in 7 of the 8 regions. And I really think that demonstrates the movement we're seeing in the expansion of this tick. It was originally detected in Southeast Nebraska in the early 90s. And you can see now we've seen it in 7 of our 8 regions. The only region we haven't found yet is in the panhandle part of the states. And if we actually look at the density itself here, so over in this far right side here, we see the tick density, the number of ticks collected per 100 meters sampled. We see that the east, central, and southeast portions of the state are, have had the highest density of the Lone Star tick collected. And that's not too surprising considering that it was, first detected here in Nebraska in that area and then has gradually moved further north and westward. So it makes sense that those two areas would have the highest density. Definitely something we need to, kind of keep, track of and keep an eye on, as we move forward here into the future, just because of that expansion that we're seeing with this tick. Moving on to the next tick here, Dermacentor variabilis. This is the American dog tick. And we see here, we've detected it in all regions of the state. Again, this isn't too surprising. We've known for a while this tick is our most widely distributed tick in the states. And if we look at that tick density, the central part of the state here really seems to be where we see the vast majority of the ticks. And again, I don't think that's too unexpected. This tick is primarily associated with more grassland and grassy habitats in this part of the state. We certainly have a plethora of grassland and grassy type habitats where this tick can be found at. And then the last tick here, Ixodes scapularis, this is that deer or black-legged tick. And from our surveillance activities, we've only detected it in two regions of the state, the Northeast and East Central. Again, if you look at the density and compare it to our other two ticks, that this density is much more lower compared to the Lone Star and American dog tick. But this, I think, still, is important to keep track of this tick. This is the tick that is responsible for things like Lyme disease, anaplasmosis, etc. And so it's something we're starting to see an incursion into Nebraska, and a reason we want to really keep track of this tick going forward. Now, if we look at tick pathogen testing, I do want to point out we haven't finalized the 2024 pathogen test results yet. So this only goes up through 2023. But we're going to look at the Lone Star and dog tick pathogen testing results here, and we're going to start off with the causative agents of ehrlichiosis, Ehrlichia chaffiensis, and Ehrlichia ewingi in these first two columns here. And really, what we can see is we have detected ehrlichiosis in our tick populations. The vast majority of those have been in Lone Star ticks, which that is to be expected. We know Lone Star ticks are the primary vector tick for ehrlichiosis, so it makes sense that we'd find most of our detections there. The interesting thing I think is that with Ehrlichia chaffiensis, we have a much lower infection rate here, so it's less than 1% compared to Ehrlichia ewingi, which again is still a low prevalence pathogen, but compared to Ehrlichia chaffiensis, it is actually much higher. And so I think that's kind of unique. I think oftentimes when we've done case investigations and with old CDC case criteria, most of our cases were classified as Ehrlichia chaffiensis. Based on this tick testing data, probably most of those were probably more likely to be Ehrlichia ewingi. It's just that, our primary tests that we see reported to us is typically going to be a serology-based lab, and that can't differentiate between these two species, unfortunately. I do think, if you look here again, East Central and Southeast Nebraska are the areas where we have the highest pathogen prevalence for these two pathogens. Again, not too unexpected, given that is where we find most of our Lone Star ticks, at least in terms of density. So it makes sense that this would probably be the area where we have our highest pathogen prevalence as well. Now, if we move over to the third column here, this is the spotted fever group, Rickettsia. So this group includes things such as Rickettsia rickettsii, that is the causative agent of Rocky Mountain spotted fever. However, I do want to point out we have actually only had one single detection of Rickettsia rickettsii in over 1,000 samples of ticks, which really does seems to really match up with what we see reported in the literature. While Rickettsia rickettsii is found in ticks, it seems to be at a very low prevalence level, and that kind of seems to be what we've seen here in Nebraska as well. But if you actually look at the pathogen prevalence rates, you see that these are much higher. In fact, they're our highest pathogen prevalence we've detected in the ticks. And these are due to other species of spotted fever group Rickettsia. Now, these other spotted fever group Rickettsia have been associated with human illnesses, albeit these illnesses have typically been much more mild in terms of severity compared to what we have seen with confirmed cases with Rickettsia rickettsii, the causative agent of Rocky Mountain spotted fever. And then finally, our last column here, Francisella tularensis, our causative agent of tularemia. You see we've detected that in 5 different regions in our ticks. Still primarily a low pathogen prevalence in the ticks that have been submitted for testing. Although I do want to point out here in the Panhandle region, we've had estimated infection rate up around 36%. But I do want to caution folks to really kind of take that with a grain of salt. We've only tested 28 ticks that have been collected from the Panhandle, so a very low sample number. And additionally, with these 10 positives, 5 of those actually came from one site that was sampled in 2023. So, that's probably like we just happened to hit the perfect spot at the perfect time when we had a lot of tularemia in that area. So I really want to caution folks, to in terms of the results presented here for the panhandle until we get a little bit more data there. I think once we get more data from the panhandle, more ticks tested, I suspect we will see this infection rate definitely go down in terms of the percent positivity. And then switching over then to our deer slash black-legged tick. Looking in the first column here, looking at our causative agent of anaplasmosis, Anaplasma phagocytophilum, you see that we've had detections of that potential pathogen in the two regions where we've detected the tick. However, some good news is that these have been non-pathogenic strains that have been detected. So the ones that have been found in these ticks are ones that are primarily found in white-tailed deer and are not known to cause any disease illness in humans. If we go over to the second column here, Borrelia burgdorferi, so this causative agent of Lyme disease, this is the one I think we need to pay attention to the most. You can see from the ticks we've submitted from the northeast part of the state, nearly 55% of those have tested positive for the bacteria that causes Lyme disease. However, with that, this has been very focal. All of these positive ticks have come from one county in particular, and that's Thurston County up in northeast Nebraska. And if folks remember, Thurston County is the county where we had our first evidence of locally transmitted Lyme disease that occurred in Nebraska. That was back in 2021. So it really seems that the sites that we are collecting ticks from in Thurston County really seems to be a hotspot for Lyme disease. And if we can compare that to East Central Nebraska, we've collected a similar number of ticks, but we have not detected any Borrelia burgdorferi from those ticks. Now, why that could be, there's a number of possibilities. Could be that maybe the host, the Lyme disease bacteria host, is just not in these areas in the East Central part of the state. Could also be because of maybe differences in tick population, and by that I mean there are two known distinct populations of deer tick. There's a northern population, which has more of a predilection for feeding on rodents that are the host for the Lyme disease bacteria. And then there's a southern population which doesn't feed as often on rodents, typically tends to feed more on things like reptiles and amphibians, or excuse me, reptiles and potentially amphibians and birds. But we don't know that yet if there's these two distinct populations. That is something we're currently working on with some folks at the UNMC to really look at those genetics, those population genetics, to see if we actually see a difference in if we're seeing two different populations occur within the state. And then finally, the last thing I want to point out, a few slides ago, I had mentioned Borrelia miyamotoi, which is a pathogen that causes hard tick relapsing fever. And as I mentioned in that slide, we can use this tick surveillance to really see, and get an idea of what the risk of exposure is for tick-borne diseases that are maybe not reportable within the state. And we have certainly found a single detection of that here with our surveillance. So this is the first detection I'm aware of this pathogen in the state of Nebraska. So again, this Borrelia miyamotoi is not reportable nationally or at the state level. So I think this is another example of how tick surveillance can really help us out in determining risk for picking up these pathogens within the state. Outside of that, the rest of these pathogens, the Borrelia mayonii, which is another bacteria that can cause a Lyme disease-like illness, Babesia microti, and another Ehrlichia species, we have not detected those yet. But again, I think this is what is good about tick surveillance is it lets us be able to help hopefully find these pathogens and be able to communicate that risk to the general public as well as to clinicians and policyholders. And with that, that's all I got. So if anyone has any questions, I'm more than happy to answer those questions. And if not, you can feel free to put those in the chat as well.
Thanks a lot, Jeff. It's always very interesting. Do we have any questions for Jeff before we move on? Okay. We'll move into our second agenda item. So we asked Lacey Pavlovsky, also with DHHS, it's the wrong screen, to talk to us, give us a little update on the CRS situation.
Hey, Ben, I actually think Dr. Ashroff is
Oh,
going to get. He's on, though. He's ready.
okay, that's great. Yep. Um,
Thanks.
E- Thank you. Thank you. Um, so the CRS situation, I, I think everybody is aware of the last year CRS cluster that there was a harm that was sent out, I think March of 2024, where we identified for the first time a transmission of Candida auris in Nebraska. There were 6 cases in total, when that was identified, 5 in 2024. One case was definitely acquired CRS outside of Nebraska, and then 5 within the state. Since then, and that was I think in March 2024 that we sent out that report. We did a containment response. We kind of communicated with everybody, and since then we have not seen the cases of CRS until this year, when we have the similar event. Someone from outside the state received healthcare in Nebraska, and then kind of led to the transmission of CRS within healthcare setting. Um, involving now involving multiple facilities that where we are kind of responding to at this moment. So there there are 6 cases that we have become aware of for this cluster, that we are still in the process of containment. There is colonization screening ongoing in many different facilities that are going on right now. We don't know whether this cluster is contained yet or not. So the reason to provide this update was to kind of make sure that everyone, is looking out for Candida auris cases. The Han that we sent out last year is still good this year too. The same recommendations that were listed last year are still applicable this year, in terms of the response, the sending the samples to NPHL when a lab is not able to identify the species for the Candida, especially if the Candida was detected from a sterile specimen, or if there's a suspicion of a misdiagnosis of a Candida species, to send it out to the NPHL to identify or rerun that isolate. I think it will we will have to keep our threshold low when we have a suspicion to get these tests done, so that we can do better containment. Last time around, we had identified in 2024, I'm talking about, we have identified 366 exposures, and we tested about 148 of those 366, to kind of contain that outbreak and rule out any additional cases. Some of them that went home and never came back to the hospital or nursing home, we really don't think them as a um, uh, high risk, so we don't necessarily do the screening on those folks again. So that's why there was not 366 out of 366. And this time around, I think our numbers for exposures are probably already getting over 100. Lacey probably will have a better idea, but there is a large number of folks that are still considered exposed in this particular outbreak. And when we say exposed, we're talking about those people who were on the same unit where a C or is confirmed case was present while receiving the healthcare. So that's basically how we define the exposure usually. So there are people around that are considered exposed that probably receiving healthcare today. So please keep an eye. And that's basically, was the main message that I wanted to give out. Ben, is there any other additional things you want to from the lab perspective want to add on that?
Yeah, I just want to remind everyone, we shared these and we can share these again. Depending on, the method you use to ID yeast in your lab, CDC has a guidance for possible misidentifications. I will actually link to that in the chat. It's on the CDC website. So I just wanted to remind everyone of that. And I think that's the big thing
Yeah,
for
and I
most of
think a
us.
link to that was also available in the Han also. As
Okay.
long as the CDC have not changed the link, um, I'm pretty sure we included that in the Han also when we sent out. But yeah, it's good to have both link available for everybody.
Yeah.
And I put a link to the Han in the chat as well.
Yeah, I think the CDC website has changed. So many of the things have moved around on the CDC website. So even though we may have put the link in the Han, it may not be up to date now. So it's good that you have already shared that, Ben.
Okay. Yep. So, all right. And is there any questions concerning CRS identification or any questions for Dr. Ostroff? Okay. Thanks. Thank you, Dr. Ostroff.
You're welcome. Lacey, you have anything else that you wanted to add? Did I miss anything?
I don't think so.
Okay,
I think
good
we're
enough.
good. Thank you.
Thank you.
Okay. And you can always reach out with questions and we'll get you to the appropriate person who can answer those for you. Let me see here. Is my screen being shared? Yeah. Okay. So letter C here, it might be known to some of you, and it is definitely known to some of you that one of the high throughput labs in the area has suffered an accident, and they've had to switch to emergency operations. And so we wanted to kind of bring this up as a case to kind of remind everyone, nudge everyone to have a look at their emergency operations and their surge planning. Some of the other labs in the area are taking on a lot of work from the lab that is down. So they are experiencing their surge operations now. So it's never a bad idea. You never know when something terrible could happen in the laboratory. So that's a little PSA, just it can happen. So kind of, yeah, have a look at your emergency operations and your surge planning. Yeah, it's quite a big deal. So are there any questions about that? And if you need resources or anything like that, feel free to reach out to me. I'd love to help in any way that I can. But yep, just a quick note on that. And then if there's nothing, we'll move on to our section updates, starting with the HAIAR team. I don't know who's going to jump in for that.
Well, I think the Canada Auris is the most pressing need for us, or pressing work that we have been doing, because as we said, there are multiple facilities that we have to reach out to make sure that we are reaching out to all the exposed individuals who are receiving healthcare in any of the facilities, to make sure that we rule them out for Canada Auris. So that is taking a lot of time from us at this moment. Apart from that, there's no unusual things to report.
Okay. Thank you, Dr. Ashraf. We'll move on to SARS-CoV-19. Is Cy on?
Hi, Brian. Yes, I'm on.
Hi.
So nothing in COVID world, everything is going normal, at least for now. So
Okay,
nothing significant to report. Thank you.
that is good. Derek with the flu, RSV and flu, respiratory.
Yeah, thanks, Ben. So, since last time we've talked, influenza activity has definitely come down quite a bit. So, the current levels, at least a test positivity perspective, we're actually around 3%. To put that in a little perspective, when we peaked in our test positivity back, I want to say in January, February, I can't recall the exact date off the top of my head, but we were just under 40% positivity during that time. So have come down quite significantly. And that 3% that we're seeing right now is pretty close to the levels we expect to see during our non-season or summer weeks. So that's good there. And just kind of looking at a bird's eye view of the season as a whole, specifically with the laboratory data, overwhelmingly saw influenza A predominate throughout the whole season. The first half, so kind of like our ascension to the peak, seemed to be more influenza A H3 driven, while that second half towards the peak and then subsequently following the peak and our decline saw a lot more influenza A H1. So overall, we've seen a pretty close to an even 50-50 split between the number of H1s and H3s identified and reported. To date, we still haven't seen a lot of influenza B activity, although after being on a few calls with some national partners from other states, some states have reported seeing a little bit of flu B activity recently, so it's not out of the realm of possibility that we could see a flu B wave yet. But at least right now, our data doesn't show that to be the case. So knock on wood, hopefully that stays the same. With RSV, we are still seeing elevated levels of RSV. It's kind of been a little unusual seasonality in that the peak that we saw from a test positivity perspective was about 15%, which that's not like a record high or anything unusual. But what was unusual is just how long that peak level was sustained. Typically, we don't see like a peak level of positivity for RSV last more than maybe a couple weeks. This season, that was not the case. That 15% level, give or take a percent or two, kind of maintained for almost 2 months. Thankfully, these past 2 to 3 weeks, we've started observing consistent declines. So preliminary data for week ending the 12th of April was about 8%. So it's definitely still elevated, but some more room to come down to where we would expect to see it over the summer, about like a percent or two. So I just wanted to call that out as well. If there's any flu or RSV related questions, please let me know. And then finally, just want to continue to say thanks to everyone for submitting specimens to the public health lab over the course of the season. We know we saw a boatload of flu, as I'm sure all of you can attest to. So really appreciate you guys' efforts again. So
I just want to speak to that real quick, Derek. Everyone, this is Emily from NPHL. As Derek hinted, we asked for you guys to increase respiratory submissions to us this year so we could get a really good handle on what was circulating throughout the state. Last year, at the end of the season, we had tested about 250 total samples for influenza subtyping to allow us to know which specific subtype of influenza was circulating. You guys really stepped up to the plate and knocked us out of the park this year. It was awesome. We did over 6,000 samples as of the end of March, and we are still going. So thank you to all of those labs that took the time to package up those samples and get them into us. We truly know what is going on in respiratory surveillance in this state, and I feel very good to know that we have an active surveillance program to catch a potential avian influenza should there have been one. So thank you again.
Definitely kept everyone busy. Thanks, Derek. Thanks, Emily. I guess that brings us to enteric outbreaks.
Hello, this is Brianna. Thankfully, I have no local or multi-state outbreaks to report at this time, but we are, slowly going into the summer months where we'll see probably several outbreaks. Salmonella, specifically backyard poultry, we see that every year around spring, so we'll be seeing that here in any time. And then also cyclospora. That season is generally May to September. We see around 20 to 30 cases a year usually. So um, we're gearing up for that and just ready to get those reports and get that stuff reported to CDC. So it seems like every year there's also cyclospora outbreaks too, usually leafy greens. So yeah, that's what we're up to.
Some things to look forward to.
Yes. I actually like cyclospora. I think it's kind of fun. Probably not fun to have, but fun to look into.
Thanks, Brianna. Jeff, do you have anything to add?
Nope, no additional things to add.
No additions. Okay, thanks. Is Kristen on for TB?
I am. The only update I have is that the MDDR or Rapid Drug Susceptibility Testing Lab at CDC is running. I know there have been possibly questions after all the changes at CDC recently, but that is an ongoing resource for anyone that does suspect drug resistance. Other than that, I don't have any updates.
Great, thank you very much. Okay, then we have the STD program, if Tammy's on. Usually, and then we'll take it to Ryan or Sandra for lead testing.
No updates from the lead program today.
Okay, great. So then we'll just talk a little bit about some NPHL side stuff. We have posted a measles collection and handling guide on the website. So that's available for reference. We see the LPX survey. I think most everyone has called that one in already, and the challenge set is out. So if you received that, remember to use the 24/7 pager to call in your responses in addition to your online responses. And then our next packaging and shipping class is being scheduled for late June. Information about sign up is going to go out within soon. So look forward to that if you have some staff or yourself who are in need of packaging and shipping training. That's all I have. So we'll give it a quick open mic, or if there's any new staff that need to be introduced, now is the time. Okay, hearing nothing.
I did have a question. Can you hear me?
Yes, I can.
This is probably to Derek. For sure, as of like the first week of May, are we going to stop submitting flus and RSVs?
So our typical surveillance season runs through the end of May, so we would like to keep that request going through then. The specific date for when our quote unquote surveillance season ends is May 24th. That's a Saturday. That also represents MMWR week 21, which is what us EPIs kind of use to keep track of the weeks throughout the year. So yeah, ideally through May, but if you have any questions on that, you can contact me at any time.
Thank you.
Yep.
Okay, any other questions? Okay, well, we can go ahead and bring this meeting to a close. Thank you, everyone, for joining us. If you're in the lab, you can go ahead and hop off, and we will see you next month, May 12th.